Okay, so once they've completed 1 and 2, you're going to go ahead and ask students to complete 3 and 4. All right, so to complete 3 and 4, students are going to obtain a thin strip of paper, and they're going to actually transcribe. Okay, so the lab tech will have cut out strips of paper. Now, if you cut the paper long ways instead of this way, it'll be a little longer. If they're using their laminated strip of DNA, one sheet of paper long ways should be enough. But if they write really big, they can tape them together. I've taped 3 together. The reason I tape 3 together is I'm going to do it up here, right? And you can demonstrate it up here after they try it first. Okay? So you can do this, and you would just lay it down, and you just write exactly what I wrote there on the board. So they're going to transcribe their DNA into RNA on a strip of paper. Now, you want to point out to them that they need to pay attention to where it's in bold. Okay? So you need to mark it somehow on their paper that this is a bold section, this is not. Maybe have them underline it or something. Okay, so that's 3. They're going to obtain a strip of paper. It's going to serve as their RNA. For number 3, it actually asks them what are the main differences between DNA and RNA. And so you're just going to go around to each table and talk about that. So we've already talked about 1. They use a U instead of a T. Okay, another one is that it's more degradable. The 2 other things that are different between them is that DNA is usually double-stranded. Oh my gosh, I can't spell double. Wow. DNA is double-stranded, and RNA is generally single-stranded. It's just a carbon copy. And then the other thing is that it's RNA instead of DNA. So DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. So their sugar is a little bit different. It just has an extra oxygen on RNA. Okay, but somehow that messes with how degradable it is and the way that the body processes it. Okay, so those are the differences between DNA and RNA. All right, so then number 4, it says to transcribe it. So they're going to transcribe the hemoglobin gene into an RNA piece, and it says mRNA. So this here, we're going to modify this. This RNA that I'm building is an mRNA. And mRNA is messenger RNA. It is the message that's coming out of the nucleus to be built by the little chefs in the cell. Okay, so that's your mRNA. Let's see. Okay, and then it says make a note of which ones are bold. Make sure as you're walking around and people are writing this, make sure of 2 things. Make sure, number 1, that they are base pairing to the bottom strand and not the top strand. So they want to end up with a sequence that should start UAU. Their sequence does not start UAU, they're doing the wrong strand. Their sequence starts AUA, they're doing the wrong strand, okay? And when you get to the protein, if their sequence says UAC, they did the wrong strand. So that's one thing you want to make sure. The other thing you want to make sure is that they're marking where it's bold. Because if they don't, they're going to have to go back and figure it out, and it just takes forever. So as you're walking around, make sure they're doing those 2 things. Okay, and then it says, in number 4, it says, what enzyme normally performs the procedure you just performed? I would encourage them to Google it. What does transcription? Okay, they can find these answers, but if not, you guys can have a discussion that the enzyme that's going to sit down and do this, so there is an enzyme that'll, sit on the DNA strand, there's my enzyme, okay, and start reading it in this direction. This enzyme is called Rna Polymerase, which makes sense because it's an enzyme, ACE means enzyme, and it's making an RNA polymer, right? So Rna polymerase is the enzyme, and then it says what else must bind to DNA to help this enzyme begin transcription? Again, have them Google it. But it turns out that RNA polymerase requires transcription factors. So we're going to also get transcription factors that are going to bind to the DNA also. There's a big blob of enzymes that are bound to the DNA. Transcription factors become important when we talk about cell signaling, because what made this cell start transcribing in the first place? Well, it's transcription factors that are signaled from other cells, or that are caused to be built by another cell's signal. These transcription factors do a lot of regulating, and that's really all we need to know about transcription factors. We'll leave the rest to lecture. So that's number 4. So they're going to build their mRNA, and then you're going to stop and have a discussion and make sure that everybody say, hey, did everybody start with a U? UAU. Oh, and they're going to end at the polyadenylation signal. So they'll end right here with an A. Now, something that I want you to emphasize, and that is that the mRNA is always identical to the code. Okay? mRNA will always equal the code, the coding strand. If your mRNA ends up looking like the template, you did it wrong. The mRNA should be an exact copy of the code, of the recipe that we want, except U's instead of T's. It's the only difference that you should see. And this is really hard for students, and it's something you should go over again and again. You should check every single student that's doing this and make sure they did the right strand, and if they didn't, correct it and tell them why it's wrong. Okay, it's literally, you should talk to every person and make sure that they understand this, because that's the biggest mistake we see students make on tests. Okay? All right, so once they've done that, then after you have a little discussion, say, okay, I want you to do numbers 5 and 6, and go. Let them go do 5 and 6. And in number 5, it's talking about exons and introns. Okay? And so, in eukaryotic genomes, there is a ton of junk. And the junk is called an intron. All right? So introns, they're called introns because they stay in the nucleus. They don't leave. These are your junk sequences. Exons are the coding sequences. And they're called exons because they exit the nucleus. Okay? So we have introns and exons. Why is there so much junk? Let me illustrate what junk is. It would be like if you read a recipe and it said, add 1 cup of flour, add 1 cup of sugar. So I went to the store the other day and I got the cutest red sweater. Add a teaspoon of salt. What? What was in there? What was that? It's just junk, and we think that evolutionarily it's probably leftovers from a lot of lysogenic viruses that have inputted their DNA, some from genome duplication that have then become silent genes. So there's a lot of explanations for why we have so much junk, but we have a ton of junk. Like, I don't think students even realize how much junk there is, and I'll illustrate it in lecture. But there's a ton of this junk, so we have to get rid of the junk. But before we do that, number 5 asks them to hypothesize why we might have so much junk. So you can go through the evolutionary hypothesis of lysogenic viruses, you can go through the hypothesis of duplicating a genome and then mutations happen and you get junk. You get jumping genes that happen, transposons. One of the other reasons, though, is alternative RNA splicing. So alternative RNA splicing is almost like a choose-your-own-adventure gene, right? So if you keep some introns, it becomes one gene. Or exons, and if you keep other exons, you can get other genes. And then, even trickier than that, is that some genes overlap. So this gene running in this direction and this gene running in this direction will actually overlap, and so it's junk in this gene, but it's not junk in this gene. So there's a lot of reasons, and students can come up with all kinds of hypotheses, like maybe it's a little safety net, so if you get mutations, it doesn't necessarily mutate the gene. Sure, we don't know all about introns yet, so their guess is fine. Yeah, so that's how much junk. What's actually doing the cutting? So they're actually going to take their strand of DNA, and they're going to cut out and re-tape together, right, their exons, so it becomes a much shorter sequence, okay? They're going to do that. So the scissors represent what we call a spliceosome, which means a cutting, splice, body, some means body. So it's a cutting body. A spliceosome, you don't need to go into detail here, it's complex of proteins. They actually recognize sequences on the beginning and ends of the introns and exons, and that helps them recognize when to cut it out, when to leave it in. But they're like little scissors that clip and re-splice DNA together. Okay, all right, and then number 6, it says there's a couple other things that have to happen to your mRNA. So they're going to take their finished mRNA and they're going to add 2 things. One thing is a 5' cap, and the tech will need to make copies of these and cut them out so there's enough for all the labs. They'll put a 5' cap on the 5' end, and they'll just tape it on, and then they'll have their piece of DNA, and on the end they'll put a poly-A tail. Okay, see that? Poly-A tail, 5' cap. They'll tape it to their little mRNA. And you can talk about these. The 5' cap helps find the ribosome. That's the little chef that's going to build the recipe. The poly-A tail helps it with not degrading too soon and a couple other things. So Don't go into detail, you can leave that for lecture, but it does get these two things. So if I asked what happens to a piece of RNA before it leaves the nucleus, there are three things. One is splicing, introns and exons. One is 5' cap, and one is poly-A tail. So those are the three things that have to occur before it can leave the nucleus to be translated. All right, I'm going to pause there to do translation.